In this video, we will learn how to find the biochemical oxygen demand or BOD of water samples using Winkler's direct method. But before we proceed, let us first define some concepts surrounding BOD. The first one is the dissolved oxygen. Dissolved oxygen is the amount of gaseous oxygen dissolved in water. Oxygen can get into the water in several ways. The first one would be through diffusion from the atmosphere. Oxygen from the atmosphere dissolves and mixes into the water's surface. Second way on how oxygen can get into the water is through the photosynthesis of algae and underwater grasses, through the release of oxygen. And the last one would be wind cycling. When water flows into the bay from streams, rivers, and the ocean waters generally have more oxygen. River waters are fast-moving, which helps oxygen from the air mix in. In the figure, we can see here the different demands of different aquatic animals for dissolved oxygen. A certain amount of dissolved oxygen is required to sustain fishes, animals, and other aquatic life. Excessive or lack of dissolved oxygen can cause negative changes in an aquatic system. Now, what is the biochemical oxygen demand? It is also called biological oxygen demand. It is a measure of the amount of dissolved oxygen required by aerobic microorganisms to decompose organic matter present in water at a certain temperature over a studied period. Normally, we study BOD levels in a span of 5 days because in that case, the microorganisms have already decomposed organic matter or completely decomposed organic matters present in the water. BOD value is usually expressed in milligrams of oxygen per liter of water. The rate of oxygen consumption is affected by a few variables: temperature, pH levels, the presence of certain kinds of microorganisms, and the type of organic and inorganic material in the water. Sources of biochemical oxygen demand include topsoil, leaves and woody debris, animal manure, effluents from pulp and paper mills, wastewater treatment plants, feedlots, and food processing plants, failing septic systems, and urban stormwater runoff. The level of BOD of a water sample indicates the level of water pollution present in it. So this is very important in the aspect of water or wastewater treatment because it indicates pollutants that is present in water samples. Process of finding BOD using Winkler's direct method. The following are the materials that we need for this experiment. We need aspirators, 50 ml beaker, 300 ml BOD bottles, 2 bottles per trial. The first one would be for the initial BOD level and the last one would be the BOD level after 5 days. Wash bottle with distilled water. 2 50 ml graduated cylinder, or we can use 1 100 ml graduated cylinder. Manganese sulfate, alkali iodide solution, concentrated sulfuric acid, starch solution, (10 ml pipettes. We will use aspirators together with pipettes. And for this experiment, we'll need 4 pipettes. We will need 1 pipette for each chemical to prevent contamination. Sodium thiosulfate, iron stand, 50 ml burette, and of course the clamp to hold the burette. And don't forget your water sample. Now moving on for the procedure of the BOD test. The first step is to place your samples in your BOD bottles. We will need to keep the other one for incubation for 5 days. And the first bottle will be directly tested for immediate dissolved oxygen levels. The first step is to place 1 ml of manganese sulfate into the water sample. Using the pipette and the aspirator. After this, we will add 1 ml of alkali iodide reagent or solution, making the addition well below the surface of the liquid to avoid inclusion of air bubbles and using pipettes. We will stopper the bottles and carefully mix by inverting the bottles several times. We can do this for about 20 times. After inverting the bottles for several times, we will allow the precipitate to settle. Now this will take a few minutes, that's why we will just fast forward the video. If white precipitate is formed, discontinue the analysis. But if a brown precipitate is formed, continue with the analysis. When about 100 ml clear supernatant has appeared above the precipitate, carefully remove the stopper. Now we add 1 ml of concentrated sulfuric acid down the side of the bottleneck using a pipette and aspirator. After this, stopper the bottles and again mix by gently inverting until the precipitate is dissolved completely. After this process, we will withdraw 100 ml of the solution in a graduated cylinder. Now since we don't have a 100 ml graduated cylinder, we will use 250 ml graduated cylinder to measure the amount of solution that will be withdrawn from the water bottles. Now for the titrating solution, we will need to place our sodium thiosulfate solution inside the burette, the 50 ml burette. After placing your sodium thiosulfate solution inside your burette, we will need to clamp the burette that is attached on the iron stand. Now after this process, we will start with our titration using sodium thiosulfate solution. We will continue with our titration until the solution starts to become of pale yellow color. This is for the 200 ml remaining sample. After titrating to a pale yellow color, discontinue titration. Now we will need your starch solution for this next process. Using a different pipette, add 1 ml of starch solution and continue titration until the blue solution becomes colorless. Now in this video, we will see that after adding the starch solution, your water sample solution starts to become color blue. After setting aside your starch solution, we will continue with the titration process. It's becoming colorless already. We will stop the titration process. After this, we will look into the amount of sodium thiosulfate that was used for the titration process. And based on this experiment, there was 6.2 ml of sodium thiosulfate that was used. Now for the second sample that was incubated after 5 days at 20 degrees Celsius, we will repeat the same process and we will record it as dissolved oxygen final. The first reading is always your dissolved oxygen initial or the immediate dissolved oxygen. Now for the computation part, the BOD level or the biochemical oxygen demand after 5 days is computed by subtracting the dissolved oxygen final from the immediate dissolved oxygen levels. And that is how you find the BOD levels of water samples. Thanks for watching. If you like this video, don't forget to hit the like and subscribe button and turn on the notification bell.